Hello friends, in continuation with the part 1 series, we are now going to see the next variance, that is dominance variance. For additive genetic variance, you can see the previous video, that is part 1. Now what is dominance variance? Dominance variance can be seen as deviation from additive genetic variance. That is, it is the result of intra-allelic interaction. Intra-allelic interaction. And additive variance is also the due to intra-allelic interaction. Both are due to intra-allelic interaction only. Here you can correct. Here they have written additive genetic variance. It is dominance variance. The result of this dominance variance is due to the deviation of heterozygote from the average of both homozygotes. Suppose you are having two homozygote, that is capital A and small a, and if you are having heterozygote, that is capital A small a, the deviation of the effect means difference in the effect of capital A small a due to the as compared to the effect of both the homozygotes, that is called dominance variance. Now the main point related to the dominance variance are, it can be of three types. Dominance variance can be of three types, that is incomplete dominance variance, complete dominance, and over dominance. Magnitude. In natural plant breeding, dominant genetic variance is always lesser than additive genetic variance. We have seen in the last video that additive genetic variance are predominant in natural breeding population. So if question is which variance is predominant in natural breeding population, it will be always additive variance. The magnitude of additive variance will be always greater. Okay. Now association. The dominant genetic variance is associated with heterozygosity, and therefore it is expected to be maximum in cross-pollinated species and minimum in self-pollinated species. The dominant genetic variance is, what is the reason for dominant genetic variance? It is due to heterozygosity. That is, if this is the result of heterozygote, and heterozygosity can be obtained when you are crossing to different species, like this is formed by crossing between capital A into small a. So dominance genetic variance is always found in cross-pollinated plant. It will be maximum in cross-pollinated plant and will be minimum in self-pollinated plant. Okay, now next is measurement. How we will measure the dominant genetic variance? So dominant genetic variance is measured by specific combining ability. Specific combining ability is used to measure. For additive genetic variance, we are using GCA. GCA is used to measure for additive genetic variance, but for dominance we are using SCA. That is SCA, specific combining ability. Now effect of selection. Dominance variation is non-fixable, and therefore selection for trait governed by dominance variance is not effective. As we have seen previously in additive genetic variance, that they are fixable, but dominant genetic variance, they are non-fixable. They cannot be fixed. Therefore, the selection for them is ineffective, because if we cannot fix them means their magnitude will be differ every time you will cross them or allow to self-pollinate. They will deviate from their magnitude will not be same as the parental population or as you have selected. so their selection is not effective. That's why we are not able to maintain heterozygote. Like if we are making F1 hybrid, we are not using the seed of F1 hybrid to sow again we cannot use the seed harvested from F1 hybrid, because due to segregation the dominance in F1 hybrid cannot be transferred to F2. They will show segregation in F2 and their magnitude cannot be fixed. So we are not using seed harvested from F1 hybrid. That's why the farmer are not using the hybrid seed, because they know every time they need to change the seed, which is costly for them. Now if where we use this dominant variance, it is used in heterosis and hybrid vigor, mainly in cross-pollinated plants. Heterosis and hybrid vigor is the phenomenon in cross-pollinated plants. So we are using mainly in heterosis and hybrid vigor dominant variance. Now another major effect of dominance variance is it calls when you are going, there is a dominant variance in any crop and you are going for selfing or inbreeding, then it will called inbreeding depression. Inbreeding depression is nothing but decrease in the magnitude of heterosis or hybrid vigor over continuous selfing. If you will self any cross-pollinated plant, then there will be loss in the high yield or vigor. This is known as inbreeding depression. Inbreeding depression is major phenomenon in cross-pollinated plant. Remember this, not in self-pollinated plant. Now we will see our third variance, that is epistatic variance. What is epistatic variance? It is the deviation from additive scheme of the gene axon which result due to interallelic interaction. We have seen now two variation before that is additive and dominance. Both are due to intraallelic. These both are due to intraallelic, means interaction between same allele of a gene. Suppose you have a gene, it have two allele, capital A and small a. So both additive and dominance variance are due to the interaction between these two, capital A and small alleles, which are of same gene. But epistatic variance is due to interaction between two different allele of two separate genes. Suppose you have two gene, capital A and capital B. So interaction between these two alleles, both are of different gene, is called epistatic variance. Now we will see main point related with epistatic gene interact and sometimes lead to new phenotype expression, as two different gene will combine, like capital A, capital B, or capital A, small capital small b. They will interact and they will definitely give new phenotype. The gene may be either dominant or recessive. Okay. Now magnitude. In natural plant breeding, epistatic variance has lowest magnitude. You will find the lowest magnitude of epistatic variance. Highest magnitude in natural breeding population is of additive genetic variance. Then we will get dominant genetic variance. Then afterwards epistatic variance. Means epistatic variance will be lowest in natural breeding population. Remember this. Component. Epistatic variance include both additive and non-additive component. Additive component also and non-additive component. We will see the types of epistatic variance. It include additive into dominance, and dominance into dominance. These are the three type of epistatic variance that are included.